Cloning is the process of taking genetic information from one living thing and creating identical copies of it. After that, the copied material is called a clone. Geneticists have cloned cells, tissues, genes and entire animals, and although this process may seem futuristic, many organisms produce clones through asexual reproduction, which nature has been doing it for millions of years.
Wow!
But how have scientists even thought of cloning? It all started back in 1938 when this German scientist called Hans Spemann who proposed a fantastical experiment to transfer one cell's nucleus into an egg without a nucleus, which is the basic method that would eventually be used in cloning. He died 3 years later after his experiment, but he was known as for his discovery of the effect now known as embryonic induction, an influence that directs the development of groups of cells into particular tissues and organs. Then, scientists started to think deeper, and take advice from Hans Spemann's idea of cloning.
I've got a question for you.
Why do scientists clone? It appears that some scientists would like to clone endangered species to protect them from extinction, and to hope that cloned cells will be used to treat serious diseases such as heart problems, diabetes and spinal injuries. Scientists would believe cloned cells may one day be used to treat a damaged heart or neurons, and hopefully with the right amount of technology, we would be able to use those cells as a benefit. In 1944, Oswald Avery, who was a Canadian medical researcher, discovered that genetic information is carried by the nucleic acids of cells. Just like Hans Spemann's fantastical experiment which eventually became the idea of cloning, the process begins by removing the nucleus from an egg cell and inserting a nucleus from the adult cell to be cloned. But there are 3 different types of artificial cloning and how scientists could create cloned cells. Gene cloning produces copies of genes or segments of DNA. Reproductive cloning produces copies of whole animals. Therapeutic cloning produces embryonic stem cells for experiments aimed at creating tissues to replace injured or diseased tissues. So far of all 3, gene cloning is the most common type of cloning done by researchers. Its future growth and development is determined by the unique genetic instructions and therefore, the clone has successfully been cloned. Let's see what scientists have cloned. Cloning animals has not been controversial. So far sheep, goats, pigs, cows, mice, rabbits, horses and cats have been cloned. But cloning is difficult since it takes the whole notion of sexual reproduction and throws it out the window. Precisely, scientists still doesn't know the answer to this for certain. And researchers will finally be able to clone monkeys for the first time. The first ever successful cloned mammal was Dolly the sheep. Ever since the first mammal has been cloned, scientists began to take on the next level and do more cloning. The first cloned mouse was successful in 2000. The first cloned buffalo was a year and it is. Not all are beneficial when it comes to cloning animals. So let's take a look at the pros and cons, shall we? Here are some pros and cons. In pros, the benefits of cloning is being able to create tissues and organs for doctors, as well as growing stem cells. Cloning may also have a possibility of bring back extinct species, but it is not confirmed. Cloning may as well treat serious disease in our world, and it is also used for generic research and organ replacement. For cons, cloning may increase in birth size or cause a dysfunction in body control, and as a result, the clone may end up being ugly. It can also cause negative effects, for example, variety of defects in vital organs, premature aging, and problems with the immune system. Human cloning could be a violation to human rights, so don't clone humans. In conclusion, human cloning can be a bad idea considering the lack of experience our scientists have in that area. But we should imagine how our modern world will be like if we make further advancement in cloning and how we can see differently from our perspectives by the power cloning. Anyways, that's the end of the video.